Hello, welcome to another screencast where this time we'll still be discussing nervous system and the membrane potential, and we've reached sort of the third installment here where you should be able to now describe what we mean by a membrane potential and then also explain how a membrane potential is established and maintained in a neuron. We're going to use a neuron as an example here. So we need to know what we mean by what is a membrane potential, how do we generate a membrane potential, and what that means in case of the neuron. So in previous screencasts, we've discussed active transport mechanisms, and the most classic example of an active transport mechanism is the sodium-potassium pump. And so this sodium-potassium pump is moving sodium ions and potassium ions, but it's moving them disequally. Right? So this sodium-potassium pump is an active transport mechanism, and what we say is that it's going to be establishing a potential. Another way of saying this is that we are going to be establishing a gradient. We're going to be establishing a difference in concentrations and in charge across a membrane of a neuron. And this is a characteristic of neurons, is that they have membrane potentials, is that they have a difference in charge across their membranes. So if we look at this, this is supposed to represent the membrane of a dendrite, say, of a neuron. This is the outside of the cell, the extracellular fluid, and this is the inside, the cytoplasm. And so a characteristic of neurons is that they have these sodium-potassium pumps, and they have quite a few of them in their membranes. Now, this sodium-potassium pump has 3 locations for sodium ions to bind to it. It also has locations for potassium ions to bind to it. And so this sodium-potassium pump will change its conformation from one to the other. And so this conformational change, right, is performed by hydrolyzing ATP. So here we see that when it's in this conformation, we have a phosphate group bound to it. When the pump is not in that conformation, notice that the phosphate group is not bound to it. So to get it to change its conformation, we need to continuously be phosphorylating and then dephosphorylating it via hydrolyzing ATP. So when it's in this conformation here, notice that we are moving 3 sodium ions to the inside of the cell. Right? And so these 3 sodium ions, sorry, strike that, they're not moving inside the cell, they're moving outside the cell. Ugh, can't believe I said that. So these 3 sodium ions here are moving out of the cell. And so what we're doing is, with the sodium-potassium pump, when it changes conformation, it dumps these 3 sodium ions on the outside of the cell. When it changes its conformation back this way, it's going to then transport 2 potassium ions inside. And so here we see them dumping off the 2 potassium ions inside. So what is this sodium-potassium pump doing? This sodium-potassium pump is moving sodium, it's moving potassium. 1. 2. It's moving sodiums out, it's moving potassiums in. 2. 3. It's moving them disequally. Notice that we have 3 sodiums going out for every 2 potassiums going in. And this sodium-potassium pump is constantly working to establish this gradient. Well, how does it establish a gradient? Well, it's obvious that it should be obvious to you that you're establishing a concentration gradient for sodium by moving the sodium out. Right? And so that's what this arrow, this yellow arrow, indicates here, is that the concentration gradient for sodium favors the sodium diffusing back across the membrane, but of course it won't because it's a positively charged ion. This arrow indicates the tendency for potassium to diffuse out of the cell because, of course, we're generating a concentration gradient for potassium in here as well. So potassium will tend to diffuse out of the cell, right? And that's the concentration gradient here. But of course it won't because it itself is also positively charged. So not only do we have a concentration gradient established, so we've got a concentration gradient established for sodium ion and for potassium ion, but the other thing that we are establishing here is an electrical gradient. there's an electrical difference across this membrane. Notice that we're pumping 3 positive things out, and then we're bringing 2 positive things back in. And so what this is establishing is that the outside of the cell is going to be slightly more positive than inside the cell, which is going to be more negative. Another way of saying negative is that it's just less positive. We're moving 3 positive out, but then putting 2 back in. Then we're putting less positive back in. That indicates that it's negative. So there's a charge difference across this membrane is how we describe this. Right? So this charge difference across the membrane, so there's a charge difference across the membrane, is referred to as a voltage. Right? The measure in charge difference is called a voltage. Right? So sodium-potassium pump, you should pause here and summarize what it is doing and where it is. Also, in the neuron, in the cell membrane of the neuron, you have sodium channels and potassium channels. Now, these sodium channels and potassium channels are specific for sodium and potassium. So here, this is a potassium channel, potassium ion channel. And this is supposed to be a sodium channel. And what you need to know about these channels is that they are in the membrane as well as the sodium-potassium pump. So over here we'd have the sodium-potassium pump, and it's again moving 3 sodium out, and then it's moving 2 potassium in. So that's happening. I mean, that's a thing that's going on. And then also in the membrane you have, of the dendrite, you have these potassium channels and these sodium channels. Turns out that these potassium channels and sodium channels can be opened or closed, right? And so they have gates on them. So we're going to indicate that these gates, that these channels are actually closed, right? And this one is closed, and that these sodium channels are closed, right? And that these sodium channels are closed, and these potassium channels are closed, and these ones are closed, and these ones are closed. But this is supposed to represent a potassium channel that is kind of always open, right? Now, since we've got this sodium-potassium pump going on over here, we're going to be generating, again, a positive charge on the outside and a negative charge on the inside. Well, since we have what's in high concentration outside the cell, you should be able to answer this question repeatedly. What's in high concentration outside the cell is sodium ion. Right? So what's the tendency for sodium to do? Sodium's in high concentration on the outside, sodium's in low concentration on the inside. So the tendency will be for sodium to diffuse back in across this membrane. But it won't. Why not? Because sodium channels are closed. Why won't it just go across here? Because it's positively charged and the membrane is impermeable to the sodium. So sodium not going anywhere. It's in high concentration on the outside. But we also then have potassium that's in high concentration on the inside and low concentration on the outside. So what's the tendency for sodium to do? Sorry, for potassium to do? The tendency is for potassium also to diffuse, but now in the opposite direction, out of the cell to the outside of the cell, from inside the cell to outside the cell. And so this is a high concentration here. But it can't, of course, because the channels are closed and because it's an ion and the membrane is impermeable to it. But you're like, wait a second, aren't there some potassium channels that are open? And the answer is yes. And so what ends up happening is that some of this potassium actually does diffuse down its gradient outside. Well, what is this doing to the membrane potential? What's this doing to the charge outside? Well, the logic says that this is a potassium ion. It's positively charged. So if it's diffusing this way, then it's going to be adding positive charge to the outside of the cell, which also contributes to this positive charge outside the cell. Now, what happens to this potassium that actually diffuses out? Will this go to equilibrium here? No, because you've got the pump over here pumping that potassium back in. Right? So this is potassium being pumped back in. And the pump must be working faster than the diffusion force here. Right? So what's happening is that the diffusion of some of these potassium channels, some of this potassium out of these leaky potassium channels, so this is a leaky one, right, and this is a gated one. Right? As the potassium diffuses down its gradient, it is contributing to that positive charge on the outside and less positive charge or negative charge on the inside. So both of these things, the sodium-potassium pump over here, the sodium-potassium pump, and the fact that you've got closed sodium channels, closed potassium channels, but some leaky potassium channels help in maintaining, establishing the resting membrane potential. And so what we're saying is that when a neuron is not conducting an action potential, it is polarized. So this resting membrane potential, resting because it's not conducting an action potential, right? And membrane potential referring to that there's a difference in charge and a difference in concentration across the membrane. Okay? So when a cell is at rest, that means it's not conducting an action potential. There's a membrane potential, meaning there's a difference in charge and concentration of these ions across the membrane. So both of these components here, the sodium-potassium pump, these channels contribute to this resting membrane potential, generating and maintaining this difference. Okay? Highly recommend taking a look at this animation, this tutorial, to help you understand here what we mean by this membrane potential. One final slide. We can measure the difference with electrodes, and we can measure this difference and see it on something known as an oscilloscope. And again, so I mentioned that the difference in charge is voltage, and here we're measuring this in millivolts, and so thousandths of a volt here. And so we have millivolts, and what we've done is that we can actually take the neuron here. Here's the axon, right? This is the nerve cell, the neuron. This is a section of it. And we put this measuring electrode. This is the measuring. And this one is the reference electrode on the outside. And we put this through the cell, and this is able to detect a charge here. And it's able, this reference electrode is on the outside of the cell. And so the difference between them is then recorded on the oscilloscope here. And we see that the difference is about 70 millivolts worth of charge. And where this negative indicates that the inside, where you're measuring, has less positive charge than the outside, right, or is negatively charged. So if you understand this, you'd be able to then predict what would happen to this line right here if I switch this. If I put the reference electrode here and I put the measuring electrode out there, what would that line look like on the oscilloscope? Okay. What if I put this, and then another question, what if I put this electrode on the outside here with this reference electrode on the outside? What would that line look like on the oscilloscope? Okay. So lots of information there about the resting membrane potential, about potentials in general, how that's established. And so what you should be able to do now is describe what we mean by a membrane potential. Right? And there are synonyms for this word potential. You should be able to sort of rattle those off. And then explain how membrane potential is established. There are two main things that help establish that membrane potential, and then how we maintain that, and what this resting membrane potential is in a neuron. Okay? Thanks for listening. Bring your questions to class.